Thank you very much for the opportunity to present today the progress of Graybug. So Graybug is a clinical stage pharmaceutical company. We work on innovative polymers that can actually be tuned to deliver drugs over a given period of time. We have polymers going up to 1 year delivery. These polymers are injected with a small gauge needle, IVT, and we have very strong IP. This technology emerged from Johns Hopkins University. We currently have 3 programs in our pipeline. We have a 6-month duration pan-VEGF inhibitor that we are currently developing in wet AMD, DME, and RVO. We have the same molecule in a once-a-year formulation, as well as a proprietary prodrug in glaucoma, which we intend to develop for 4 to 6 month durations. We are backed by a very strong group of investors. So our proprietary nature basically goes down to 3 innovative aspects of our technology. The first one is to make these PLGA microparticles non-inflammatory. As you know, PLGA is a very good polymer that delivers drug pretty well, but historically in the eye has created some issues with inflammation. So we have a technology allowing these microparticles not to create inflammation when injected. The second layer of technology is to make these microparticles we inject in the eye aggregate together and form a depot that stays in the eye and eludes the drug over time. And the third layer of innovation, of course, is to work on very extended duration of treatment. So sunitinib was selected because it's a very interesting molecule. First is pan-VEGF, and as Emmet mentioned in his introduction, there is a lot of evidence showing now that blocking more than VEGF-A and B can actually provide additional benefits. And as you know, sunitinib binds to all VEGF receptors, blocking as well VEGF-C and D. The other interesting fact about sunitinib is a very potent neuroprotector, and these neuroprotective properties are actually due to this very strong DLK inhibition. So we currently have an optimized formulation and preparation of our lead asset, GB102. It presents this way. It's a vial of lyophilized microparticles and a prefilled syringe of diluent. And at the time of administration, the diluent is injected in the vial, vortexed, and you extract the 50 microliters required for injection. And here is a video of an injection in a glass eye. So it's like a typical IVT injection, except the speed, which we are asking our clinicians to basically inject over 5 seconds. And you can see here the depot starts aggregating immediately when it gets out of the needle in contact with the vitreous at 37 degrees. And the depot will cure very quickly and stay nicely at the bottom of the eye. So this is how it looks like. This is a rabbit model on the day of injection. So you see the depot nicely formed. And in that particular model, it was gone after 100 days. There is nothing left behind. The polymer is totally hydrolyzed, and the patient or the rabbit is ready for reinjection. So we conducted a phase 1 study, which concluded in January this year. It was on switch patients. So these patients were all previously treated patients with short-acting anti-VEGF. They had been diagnosed not more than 18 months before being enrolled in the study, and they had to receive at least 3 injections of anti-VEGF. And they had received, in average, 5 short-acting anti-VEGF shots before being enrolled. They had to show a response to anti-VEGF, but they had also to show disease activity at the time of enrollment as assessed by fluid presence in the OCT. And these patients were given either a dose of 0.5 milligram of GB102, or 0.5, 1 or 2 milligram, and they were followed for 8 months, and we had 8 patients per arm. The primary was, of course, safety, but we looked at BCVA, CST, and time to rescue. And as expected, what we observed was this very strong maintenance of the benefits acquired by the previous injections of short-acting anti-VEGF, and this was maintained for the patients which you have on the orange line all the way to 8 months. The green line is actually the intention to treat, so it's all patients, patients without rescue or patients with rescue together. And you can see the benefits on BCVA, but also on CST were maintained over a long period of time. So of interest is, of course, the survival curve for rescue. So if you put all the cohorts together, we brought around 88% of patients to 3 months without rescue, 68% of patients to 6 months, and 42% to 8 months. But our best performing dose brought 90% of the patients without rescue to 6 months and 60% to 8 months without any major safety signal, no dose-limiting toxicity, and no inflammation reported. So we embarked into 2 clinical trials currently enrolling. The first one is a trial in macular edema secondary to DME and RVO. The patients will be also switch patients, patients having been previously on short-acting anti-VEGF. They will be given either 1 milligram or 2 milligram of GB102, and they will be followed monthly for 6 months. And this study is currently enrolling. We already have 14 patients enrolled. The readout will be Q2 next year. And we have a larger phase 2b study in wet AMD, a 1-year study where the patients will be given GB102 on day 1 and at 6 months, and it will be compared to Eylea given as per label every other month. That readout will be around Q3 2021. So these are the first pictures of the patients in the open-label macular edema study, and you see the depots are forming extremely well with this optimized formulation, and you have, you see, 3 different patients showing the depot on the day of injection. And these patients are going to be tracked for 6 months, and pictures will be taken at every single time point. So to conclude, this is our pipeline. So we detailed today GB102, which is our lead asset, but we have also a program in glaucoma, GB401, for which we intend to go into clinic next year. Thank you.